Alright, hello everyone. So in this presentation I'm going to describe the possible roles of the proprotein convertase KPC1 and the transcription factor EOR1 in undermining the novel cell death paradigm compartmentalized cell elimination. So firstly, programmed cell death is a vital process in overall organismal homeostasis. Specifically, it guides tissue sculpting during development, and it also undermines some tissue trauma responses. So during development, the hand is webbed in the fetal embryo, and it turns into kind of the normal hand shape that we have. Through programmed cell death, the webbed tissues undergo programmed cell death. And so programmed cell death is important for guidance of tissue sculpting. And programmed cell death is also important for trauma responses. So after trauma, neuron cells undergo a process called Wallerian degeneration, which is a type of programmed cell death which occurs in neurons in response to trauma. And so programmed cell death is critically important for tissue homeostasis and may undermine developmental and regulatory phenomena that may go awry in certain genetic conditions as well as cancer. So our lab is very interested in programmed cell death, and specifically how programmed cell death occurs in so-called morphologically complex cells. We know that morphological diversity of cells lends to elusive cell death patterns. And this is because morphological complexity, or in morphologically complex cells, it's poorly understood how these cells die and how their corpses are eliminated. And so we study programmed cell death in the morphologically complex cell of the tail spike cell in C. elegans, the C. elegans model system. And so we know that the C. elegans tail spike cell undergoes a novel cell death process called compartmentalized cell elimination, in which the cell dies in a tripartite way. So there are three distinct compartments, and each of these compartments die in a stereotypic way. The soma rounds, and there's the soma that rounds, the process which beads, and the distal compartment which retracts. And it was found through a forward genetic screen that one of these mutants from the forward genetic screen exhibits a very rounded cell body, or soma. And so we set about trying to elucidate the genes and proteins responsible for this soma cell rounding, because it may help us better understand how the soma dies through this CCE process. And it was found that KPC1 and EOR1 loss-of-function mutants both phenocopy modestly the NS957 phenotype, and you can see here the rounded soma in KPC1 loss-of-function mutants as well as in EOR1 loss-of-function mutants. Both have the same rounded soma as in the NS957 forward genetic screen. And it was also found in KPC1 loss-of-function mutants that the embryonic tail spike cell exhibits some soma process junction thickening. And so you can see here the normal tail spike cell soma process junction, and here in the KPC1 loss-of-function mutant that there is some thickening around the soma process junction. And it was also found that tail spike cell persistence drastically increases at 25 degrees Celsius as opposed to normal developmental temperatures in C. elegans, which is 20 C. And this was found across all of the genotypes studied here. The NS957 mutant, KPC1, and EOR1 all exhibit higher phenotypes at 25 C as opposed to 20 C. And you can see that we also tested some other temperatures as well, including 28 C. And all of this shows that tail spike cell persistence generally increases with increasing temperatures throughout this specific genotype studied here.